I'm Mohammed Bentires Alj. People call me Momo. I'm group leader at the Friedrich Miescher Institute in Basel. My lab focuses on the cellular and molecular mechanisms controlling normal and neoplastic breast stem cells, resistance to targeted therapy, and progression to metastasis. I'm pleased to welcome you to conversations with key figures in mammary gland biology and breast cancer.
I'm Rob Clark from the University of Manchester and very interested in mammary gland biology and stem cells.
I'm Gil Smith from the National Cancer Institute in Bethesda, Maryland, and I would like to think that I know a lot about stem cells in the mammary gland.
Okay, thank you. Introduction's over. So I have some questions for you, Gil, that will hopefully explore your career and your interest in mammary gland and the possibility that it might contain stem cells, which have been alluded to in your work. I guess I was going to start with the way that we met in 1999 that we had spoken about earlier, which was at the Mammary Gland Biology Gordon Conference at New England College in Henniker, New Hampshire. I remember feeling that I was immediately welcomed into the mammary gland biology community. And I wondered whether you remember your first mammary gland Gordon conference, and what can you tell us about this meeting in our field and how it contributes?
Well, I think the first one I attended was 1981. And in that particular conference, there wasn't a lot of cancer, and there wasn't a lot about stem cells. There was a lot of things about dairy farms, goats and cows, and which was okay with me because I was raised on a dairy farm. So.
Okay, and so you were born and brought up on a dairy farm, and so can you remember specifically how being involved in animal husbandry made you interested in biology, or is that not the case?
Well, I was always a curious boy. In fact, my mother used to make sure that I took a nap every day so that she would have a break. And so I think I was 7 years old when I started working on the farm actively and helping my father milk the cows and feed them. And there's a lot of responsibility because you take care of the animals. Sure. And so I was always interested in biology.
And were there family or friends who were involved in academia, or was that just a route that you took as an individual?
Well, I was the first in my family to go to a 4-year college. My maternal grandfather was a professor of agronomy in Morrisville, New York. So all of those children, and there were 9 of them on my mother's side, went to at least to 2 years of college.
So were you influenced by this family member, or were you also influenced by scientific figures in the public media?
To be very honest, I didn't want to go away from home. I was 17 years old, my whole freshman year away from home, but I earned an academic scholarship at Hartwick College in Oneonta, New York. So I went there and took pre-med, and the rest is history.
So how far from home was that? Was that a local college, a state college?
It was about 160 miles from home, so it was about 3 hours of driving, 3-4 hours.
So you say you didn't really want to go to college, but you had a desire or a curiosity in academic science?
Yes, I wanted to go to community college first and grow up a little bit and then go on to school. And there was a possibility for me to do that and then go to Cornell, but I ended up going to Hartwick College because of my academic scholarship. The end of my 4 years at Hartwick, I took the Princeton graduate record exam for biology, and I scored in the 90th percentile. And that's the reason I think I was able to go later on to Brown University College.
So 4 years at Hartwick,
And then
you
I
take
went,
your biology exam that enables you to go on to...
I didn't immediately go.
Okay.
I went home. My father had the idea that I should work on the farm and help send my 2 siblings through college. My mother said he should do, he should work in the area he wants to work in. So I was very fortunate. I got a job in cancer research at Sloan Kettering Institute for Cancer Research as a biotechnician. And my first job was to work on the problem of viruses in human cancer. And so my job what was entailed in my job was to perform cell-free extracts and viral filtrates from human tumors, inoculated them into newborn mice, and then check the mice as the mice grew up. And one of the things I learned from this was that I could passage mammary tumors or leukemia and lymphoma through the filtrate, so they must be virus induced.
So then that influenced your choice of graduate program because of what you learned at Sloan Kettering?
Exactly. I worked there for 20 months. I became married. My wife was certain to have children in a very short time, so I went to Dr. Donald Clark at Sloan Kettering and asked him about graduate schools. He picked up the phone and called up 3 very good schools: University of Michigan, Cornell University, and Brown University. Got someone at Brown University and he handed me the phone. And I went to Brown University that weekend to talk to Professor J. Walter Wilson. He arrived late to my interview. He said he was an old man. He had 1 eye, 1 lung, and 1 kidney. He was out of breath, and he said, "Start talking, and when I catch my breath, I'll ask you a few questions." That was my interview.
And was that an immediate transfer into mammary gland biology research, or was it more?
Well, there were his interest was primarily in liver, but they had 2 C3H sublines of mice and one had stopped developing mammary cancer. So they asked me if I was interested in trying to find out what happened in that strain of mice with respect to its resistance to breast cancer. And that's how I got started, and I spent my time in the library learning as much as I could about mammary cancer in mice, inbred strains, and so forth.
So did your first mammary gland biology paper come from these graduate studies, or?
Yes, my thesis was on the disappearance of breast cancer or mammary cancer in the C3H subline strong Wilson. And I published that in the Journal of National Cancer Institute. Actually, my thesis was read by scientists at the National Cancer Institute because no one at Brown was conversant with this particular area. Okay. And so my thesis was approved by the scientists at the Cancer Institute.
And is that what led you to start a career at NCI? Or
well, it was very important, but it wasn't the only thing. The most important thing, probably the best offer I had was from Johns Hopkins, but I couldn't imagine working at Johns Hopkins in the hospital because it was like an armed fortress, and I didn't think I wanted to work in an armed fortress.
What do you mean by an armed fortress?
Well, there were 12-foot high fences topped with barbed wire around the parking lot at Johns Hopkins Medical College. Not the Homewood
to keep
campus.
the patients in, or the
well, well, I'm not sure, but I just couldn't imagine being a country boy and being raised on a farm. I couldn't imagine going to a downtown facility that was fenced in. So I was offered a job in several places. I decided to start as a staff fellow in research at the National Cancer Institute under A.J. Dalton, who I knew from my cell biology studies was very important in the classical paper which described the Golgi apparatus as a cellular organelle. And he was at the time studying viral ultrastructure and trying to prove that there were viruses in human cancers. And that
seemed to be a very hot topic, if you like, at that period of time. So tell us what were seen as the major questions in the field when you started?
Well, when I was a graduate student, the issue was the milk agent. There were three factors important in the development of mammary cancer in mice: hormones, genetics, and virus. And the milk agent wasn't really known to be a virus at the time. And I was interested because the virus did not kill the cells. On the contrary, it made the cells more robust and last longer and turn into cancer. So I thought by studying the virus, I would have some insight into how cancer was formed in mammary epithelial cells.
So I get the impression that at that time, it was almost because of the ability to, because of the knowledge about DNA and the new knowledge about viruses, that it was almost seen as the cause of human cancer, and that was reflected in some of these models. How do you think that has played out over time?
Well, I think that it was clear that the special virus cancer program at the Cancer Institute was in full swing at the time I started at the Cancer Institute. And there were some scientists who believed that, in fact, all cancers were caused by a particular virus, and by immunizing humans against some portion of this particular virus, which was the C-type virus, they would be protected from developing. So it was a prevention idea. So
you could argue that in, I guess, in liver cancer and cervical cancer, that notion, that idea has been exploited, but for other cancers, it's really not been the causative agent.
That's correct. There is some evidence for the human tumor T-cell leukemia virus, which is interesting, but hasn't turned out. And of course, HIV, what is a retrovirus, and the fact that many people at the Cancer Institute and around the country had studied retrovirology was very important in the early advances that were made when HIV showed up.
Yeah, I agree. I mean, I think the virology field has, I mean, despite the fact it may not be the causative agent in cancer, I mean, it's really led people's knowledge of viruses on. So what were the other major challenges and questions in mammary gland biology and cancer?
My major instrument my major approach was ultrastructure, so electron microscopy. That's the expertise of A.J. Dalton, and so I was in an ultrastructural research facility, and I used the electron microscope. And I followed my mentor's approach, and that is I embedded tissues, tumor tissue, mammary tumor tissue, in water miscible plastic, cut ultra-thin sections, and floated them on enzymes. And by doing this, I showed that the intracellular A-type particle had a ribonucleoprotein component, and this was also present in the viral particles that were called B particles that were subsequently shown to be the mammary tumor virus.
So in fact this was the ultra-structural identification of the mammary tumor virus.
That's correct. That was published in Cancer Research the year after I got to...
So I guess that was a key discovery in mammary gland biology, in the sense that the MMTV LTR and promoters have been used throughout the period onwards for discovery of new genes in breast cancer.
I thought another thing that was very important was I was the mentor of Herbert Abelson of the C. Abel virus. So I had the opportunity to train Herb as a as a scientist. He was a physician,
right? A
very good pediatric physician, and together we identified the nuclear annulus pore relationship in sections by serial sections and Markham rotation, and we drew a model and proposed that it was a cellular organelle, and this model today is the same as the one we proposed in 1970 and published in a now defunct journal called the Journal of Ultrastructure Research.
Yeah, I mean there's a lot less ultrastructure done these days. So your mentors at NIH were the ultrastructural biologists. Were there other very influential people?
Well, Howard Andervant and the group that was in the laboratory of biology. There was Michael Potter, who came over from Building 8. We were located in Building 6. There was Lloyd Law. There was Clyde Daw, who was a developmental biologist. There was Terry Sakakura, who was there with Clyde Daw for I mean, it was a very academic setting at the time that I was there, and I learned a lot from interacting with these people at Monday seminars, Monday noontime seminars, and having lunch with them in a very informal setting. So it was really great for me as a young person to actually hear the stories about the non-infectious virus, which was called the nodule-inducing virus, which looked exactly like the milk agent, but it was and it caused tumors in animals fostered-free of the especially C3H animals. And so this was a real problem. What is this? I mean, you have a virus that looks, I mean, a virus particle that looks the same as the milk agent, but it doesn't infect and it doesn't transform, as far as we know, except in the animals that inherit it through the genome.
Yeah, I think I like the way that you talk about all the people around you at NIH, and how over lunch is actually where you discuss the discoveries that were made. You learn a lot from that experience. But in my career, I've always felt that meeting people from outside your institution and taking their ideas and taking those back with you to your own institution is important.
Even
so
outside field.
Well, of course,
outside your own institute.
Yeah.
So.
I was invited by Bob Cardiff and Phyllis Blair to speak at Berkeley, California, and that was a wonderful experience because everybody in Ken DeYoung's lab was interested in mammary cancer in mice, and from many different aspects. I mean, ultrastructure, hormones, endocrinology, immunology. It was a multifaceted group, and I learned an awful lot from being there. And the best part was I not only got to give my talk, that they invited me for, but I also got to give my talk from my thesis work, and they were actually interested, and it was wonderful.
I mean, that is amazing when you connect with those people who share the same interests, and I mean, you know, that's continued over, I guess, over 40 years, because we're here in Galveston, and Bob Cardiff is here, Dan Medina is here, we're talking about Charles Daniel and Lindsay Hinkman from his lab, so I mean the dynasty continues.
I was also invited to Baylor College of Medicine, and I met Jeff when he was a very young man, and he was studying casein gene regulation, and he wasn't so interested in the biology of the mammary gland as he was in the biochemistry of the regulation of the casein gene, but he has developed a big interest in the biology of the mammary gland as well as the regulation of genes.
Yes.
And so I met Dan and Larry Young, who was a very important person in the Cancer Research Genetics Lab at Berkeley, because he was really the hands that developed the clearing of the mammary fat pad and transplantation of tissue and cells into the mammary fat pad. And Charles Daniel and Dan Medina were both graduate students at the time Larry was there, and then subsequently Larry went to work with Bob Cardiff. So Larry basically is to Bob Cardiff as Francis is to Dan Medina.
Yeah. So one of the questions we wanted to ask you was the cleared fat pad assay that they were using. When did you first use that and apply that? Did you learn it from them, or you were?
Well, I did. Well, it became clear that electron microscopy was a technique.
Sure.
And to learn more about the biology of the gland and the interaction of the virus with the cells in the gland, one had to develop new methods for looking at it. So I was very excited to begin with to work with Barbara Vonderhaar and Yale Topper on explanted normal mammary glands that could be induced to differentiate in fragment cultures. And I was the cell biologist who looked at these cells, and I was interested in knowing whether these cells would be able to grow and produce glands. And so I learned the technique of
who taught you this technique? Was it Larry?
Larry Young was very instrumental in teaching me to clear the fat pad. He sent me the protocol, and I struck out on my own. I don't mean strike out, I mean I started out on my own, but Larry came and looked over my shoulder and helped me. I started working with check mice, which are about as big as my little finger at 3 weeks.
Probably
so it was very difficult, technically, very challenging to clear their mammary fat pads, but it was actually very easy to find hyperplastic lesions because they have very little fat.
So what was your question at this point that you were testing with the transplantation assay?
My question was trying to find out what was different about the hyperplastic lesions that were caused by the virus that were clearly pre-malignant lesions that developed tumors. The interesting thing was that the tumors developed in individual sites within these outgrowths. The outgrowths clearly were not normal. They were pregnancy outgrowths in a non-pregnant animal. So I thought pretty certainly they must be able to make their own supporting growth factors and so forth. And Dan and I worked together on this and found that, and Barbara Vonderhaar also was involved, and we found out that actually the distal glands that were normal often were stimulated to act like the hyperplasia that you transplanted into the cleared fat bed.
Okay.
And we never figured out exactly how to approach this problem, but it was very interesting and indicated that there were humoral factors that were released from the pre-malignant tissue, which affected the normal tissue in the animal carrying the pre-malignancy.
So was this methodology was the thing that enabled you to strike out on your own and have your own lab? Is that how?
I always had my one of the things about being a dairy farmer or a farmer is that you learn to do to be independent of other influences of other people. And so the thing that was most appealing to me about being a research scientist was the fact that I could do as I wished to do, and without outside interference. And I could study, and I could ask questions that I was responsible for. And I think it's important for scientists to understand that that's the best gift of being a scientist.
Yeah, I agree. I mean, so it's not so much about being given your own lab, it's about having your own independence. But at some point, NIH must have assessed you and said, right, now you have tenure now, you know. Or,
I was very fortunate because after 2 years I was converted to a permanent.
Okay.
That was short.
Yeah, very short.
And so
I didn't intend to stay there. I intended to go back to academics, but then I realized that if I went back to academics, I would, and I interviewed in a number of places, I was told my salary would be smaller, and any increase in salary would be dependent upon writing a grant which was approved by my peers. When I was at the NIH and NCI, I had a hard salary, and I had money, whatever the lab chief decided to give me, to do my research. We never had a budget at NIH as a principal investigator until 1997, and I started in 1965.
So I was going to ask what was your first grant about? But actually, you have your own independent position. You have facilities. You have money. The
only shortcomings were space.
Okay.
You could have equipment, but you had to have space to put it in. And so space was the limiting factor, and support personnel. Yeah. I mean, I didn't have an army of graduate or undergraduates like Lindsay Ing.
Right.
So I did a lot of the things on my,